Hello, everyone, and thank you for your interest in today's ICTR Enrichment Series presentation, Biostatistics, Why, When, and How. Before we begin, I would like to review a few items. Today's presentation is recorded. The recording and the PowerPoint will be posted on the ICTR website, and that link is posted in the chat feature as well as a link to an ICTR satisfaction survey. We invite you to take a few moments today to complete the survey and let us know how we can improve our presentations and what additional topics you're interested in. We will reserve the last 10 to 15 minutes or so for a Q&A session. Please add your questions into the Q&A feature as they come to mind. This feature should appear by hovering your cursor near the bottom of your screen. Now let me introduce our guest speaker, Dr. Soren Bentzen. Here at the University of Maryland, Dr. Bentzen is a professor of epidemiology and public health, as well as the director of the Division of Biostatistics and Bioinformatics and director of the Biostatistics Shared Service Greenbaum Comprehensive Cancer Center. Dr. Bentzen is also the director of the UMB ICTR Biostatistics Core and the director of the Translational Research Proton Therapy Alliance. Dr. Bentzen, thank you so much for taking time out of your schedule to present for us today. And I'm going to turn over the presentation to you if you'd like to share your PowerPoint.
Okay, thank you very much, Wanda. And it's a great pleasure to be here today and to give this lecture. And thanks to everybody who's joining in. I thought that I would start by just asking the rhetorical question, why in the world would anyone want to actually talk with a biostatistician? And I appreciate that our audience is probably a biased sample, so maybe you have an idea already. But it is a fact that biostatistics expertise is probably more in demand now than at any other point in time during my own career. And then if I succeed in convincing you that you should speak to a biostatistician, when ideally would you like to involve the statistician in the lifeline of a biomedical research project? And towards the end of the presentation today, I'll spend just a few minutes addressing how exactly do you get access to biostatistics support in the UMB environment. Now, this is from the US Bureau of Labor Statistics, a report in 2017. And it's basically the 10 fastest growing jobs predicted to grow from 16 to 2026. And you can see that a lot of these are exactly as expected, wind turbines, solar cells, and so on, home health aides and physician's assistants. But down here you have statisticians and mathematicians. And in terms of the absolute growth in numbers of jobs, it's relatively modest compared to many of the others. But the rate of growth is very steep, around 30% per year. This is from Forbes magazine. They had a feature on the 10 best master's degrees currently. And again, of course, it put a smile on my face that at the very top of that list, you had a master's degree in biostatistics. And of course, this is all about the digital age. The digital age officially began in 2002. And then you can ask why 2002? That was the point in time where half of the stored information in the world was stored on digital media. And you can see this is a review from Science back in 2011. You can see that just 9 years later, actually 94% of all data were stored in digital form. And that is such a change. Way back, and way back now means just back in the 80s, digital media were almost non-existing. Most of the data we stored were stored on analog media, on tapes or on films or on paper. And the digital age obviously gives you a completely new way to access data. They become really accessible, at least in theory, at the touch of a button. And that has opened some enormous possibilities. Maybe I should also mention that currently the rate of growth of the stored data in the world is mind-boggling. It is estimated that of the data that we have stored at this very moment, 90% has been stored within the last 2 years. So this is a very short doubling time, and we can all appreciate how such an exponential growth curve will go as we move forward. And healthcare is right in the middle of all this, obviously. And I don't think you need to be a prophet or anything to just make the bold statement that the future of healthcare is big data. This is going to dominate the field, both in terms of management of disease, management of hospitals. The economy side obviously benefits from the ability to access and analyze big data sets. And it's very much going to also set the agenda for the kind of research we will do in the future. And this is not a predicting that all other kinds of research will disappear. I personally believe we will still do traditional hypothesis-driven research also in 10 and 15 years from now. But this is really opening the gates to a whole new era of data science. And we get data from many sources, from electronic medical records, of course, from hospital and clinical information systems, from third-party databases, insurance companies, especially in this country. Lab data are generally stored in digital databases. Images are being stored in PACS systems. And also we can go out in society, and we have a group at the School of Pharmacy who is looking at whether, for instance, patients actually fill their prescriptions after they are sent back from hospital to their own home. And this comes with, again, a number of tools. I won't go into the details of this. You're probably familiar with at least some of them. Tools that allow you to combine data sets and to operate on the data sets. Out in the surrounding world, we have enormous amounts of data that are being stored based on web activity and also from social media. And again, it's easy to see the opportunities involved in trying, for instance, to mine data from social media on, say, health literacy or health issues, and try to combine that with knowledge about whether a person themselves or their relatives have a given disease, and so on and so forth. And all of this then again is the output is being used in a number of different contexts. There's a lot of interest obviously in data science per se, but there's also a lot of interest in management of healthcare, and there's a lot of interest in clinical decision support systems, both with interfaces that appeal to the healthcare providers, but also to the patients and relatives themselves. So all of this seriously was just a big future dream not so many years ago, and the future is already here. This is the kind of environment that is opening up. The future is here. It's not evenly distributed yet. Some of these tools are less accessible than hopefully they will be in just a few years, but it's really coming very fast. In healthcare and in health data science, this means that suddenly we have access to data sets that we never had access to in the past. And it basically covers the whole range from population level databases, and I'll show a few examples of that in the next slide, hospital database systems, People still collect, obviously, case series. And at the other extreme, we get very, very rich information if we take samples and if we go to, say, the Institute of Genome Sciences or whoever we work with and start to use the high throughput, high density assays. Big data is a bit of a buzzword, and you will find big data at the two extremes. Some of the population registries, we would call that big data. And at the other end of the scale, most of these high throughput omics data sets qualify to what we call big data. Now, it's important to distinguish between the two, not just because of what they are, but also the kind of statistical tools and data analysis you would use for analyzing the data sets. The kind of challenges you are facing will depend on whether you have big data at a population database level or at an omics data level. So if you have a number of cases, and we call that N here, and if P is the number of features or covariates or clinical characteristics, lab values, images, whatever, per case, then a total data set is one where the number of cases is very large compared to the number of data items per case. And down at the other extreme, we have wide data sets. And by wide, we mean that the number of features is very large compared to the number of cases. So this could be, for instance, the Sentinel, FDA Sentinel database, where you have hundreds of millions years of observation time, but with relatively few data items per case. And at the other extreme, of course, we have series where we do next generation sequencing or some other high throughput assay, but often because of the cost involved in generating these data sets, we would be looking at data sets where perhaps we have this information from in the order of 10 to a few hundred individuals, not thousands or millions of individuals. These are some of the total data sets. We just mentioned the FDA Sentinel. It's a distributed database. It means that all the data is not residing on the same physical server, but it's basically a network of servers that is connected so that you can get access to all the data when you do your analysis. And there are currently more than 300 million person years of unduplicated, curated data that are basically quality assured and ready to go, at least in principle. We also lucky in the state to have the CRISP data set. This is statewide, and it includes also a few of the hospitals in neighboring states. And again, this database stores hospitalization related information and currently contain information on more than 17 million unique patients. A lot of my own research is in the field of cancer research. We have the NCI SEER database. More than 9 million patients with cancer have fairly detailed information stored in that data set. And we also often analyze data that are extracted from the National Cancer Database, a terrific resource where you have data on more than 34 million unique cancer cases. So if you analyze data from a fairly common cancer, like say breast cancer, you are seriously looking at a data set with more than 2 million verified breast cancer data. And again, there's fairly good information, fairly good width of data or depth of data, if you like, in terms of treatment details. The big limitation to that particular database, for those of you who may have an interest, is that the outcome is only overall survival and immediate post-operative mortality. So we don't have cause-specific survival in the data set, and we don't have time to progression or time to recurrence, which obviously is a big limitation. But again, these tools or these data sets, it's all about understanding the limitations and then realizing exactly what kind of research they will allow you to do. And that's another characteristic of where we are currently. The big numbers in themselves will no longer impress people. Not so long time ago, analyzing 1 million cases of something was of interest in itself. Now the focus is more and more shifting to what exactly are the questions you can now address? What is the research you can do that take advantage of the data set, but it's not the data set per se that creates the buzz. So that's just setting the scene. What biostatisticians can do is to help you navigate the data sets, come up with refining your research questions. The research is still yours, of course, but we are able to help you formulating questions, realizing to what extent these questions are likely to be addressable in a given data set. We can, at least to some extent, help you opening the box with all the wonderful data, or we can point you to people who can help you. And again, we can basically follow you all the way from the conception of a study all the way to final publication. Now, when I give a talk like the one today, I like to look at this hierarchy of sciences, just to remind you that mathematics, logic, and statistics are the formal sciences. If you subscribe to Western science, these are the rules that you cannot break. All other sciences will have to respect the rules of logic and mathematics and statistics. You can develop formal sciences without having an ontology. You don't need a reality to develop a mathematical theorem. It's self-contained, basically. If you have an ontology, then that maps elements in your theory to elements in the real world. And that's where physics comes in. That's basically studying the world around us using mathematical principles. And chemistry and biology comes at the next level. They have to respect the laws of physics. You can see them as disciplines within physics, if you like. And then at the bottom of this pyramid, we have the applied sciences, engineering and medicine and so forth. And again, they work within the framework or the hierarchy defined by the other sciences. So how about biostatistics then? Biostatistics is kind of interesting in the context because it spans this whole range. It's all about numbers, of course. It's a quantitative science, if you like. It's a data science. Biostatistics is rooted in the formal science of statistics, but the numbers are numbers in a context. And it's important to understand that context. The whole flavor of biostatistics, if we pride ourselves of anything, it is that we try to meet our users halfway. We try to understand the biomedical setting, the experiment, the biological system, or the clinical scenario that the data are derived from. And then we try to use our basis in computational methods, in study designs, in data analysis, and so forth, to match the context, the domain of application, to match that with the most appropriate design, if it's a study design, or analytical methods, if it's a data analysis. And then after designing a study, collecting the data and analyzing the data, we can also help you with the interpretation of study findings and publishing them. One of my favorite quotes from Mark Twain: "It ain't what you don't know that gets you into trouble. It's what you know for sure that just ain't so." And that's really the flavor. I'm not claiming that biostatisticians are more clever than anyone else, but what biostatisticians are trained to do is to make conclusions in the presence of uncertainty and to critically examine what we know and what we don't know. As you're probably aware, human beings have a lot of cognitive biases that gave us an evolutionary advantage throughout evolution. And these were useful traits when we were running around on the savannah in East Africa. But if you go into a field like biomedical science, it turns out that these biases actually make you make wrong conclusions. And it's not just that people make irrational conclusions from data. They actually tend to be irrational in the same direction, if you know what I mean. And again, if you are trained in biostatistics, and of course, some of that training is on-the-job training as you work in the field, then you become really aware of these biases. And we all try to hone those skills and be better and better at revealing our own biases and trying to challenge what we know and what we don't know, and trying to come up with study designs that will really have an audience. An interesting project is obviously something you would like to do, hopefully, but also where you know there's an audience out there, somebody who would really care to know the answer to your research question. Those kind of problems are obviously the most fun to work on. Now let me show you an example, and I'm showing you this example. It's a study I was involved in when I still worked at University College London back in Britain. The setting was we looked at brachytherapy for cancer of the uterine cervix. Because of the anatomical location of the tumor, for more than 120 years, it has been treated with radioactive sources that are inserted into the uterus and the cervix itself. And that turns out in those cases that are not surgically operable, either technically or medically operable, it turns out to be actually a fairly effective therapy with a curative potential, even in fairly advanced disease. It's also a strong medicine, so it comes with the risk of fairly severe side effects, and some of those side effects are chronic side effects that will affect the woman for many years into the future, maybe for the rest of her life. Now, back in 2002, we completed an audit of all the hospitals in the UK that treated this disease with radioactive sources, intracavitary brachytherapy, as it's called. And everybody played along. Normally, hospitals are not super happy to tell about their complications, but these were seriously all the departments in the UK who delivered this kind of treatment to women with this disease. So the setting was that we took all cases in 1993 and we asked them to follow the cases for 5 years and then to report how many severe late side effects did they observe. And in addition to this, of course, we also compared the survival outcome between the departments. And it turned out that there was a wide range of complication rates in the centers participating, all the way from departments that never saw, or at least not in 1993, when they followed up these cases, they didn't see a single severe side effect. And at the other extreme, we had departments in the UK reporting 2/3 of their patients having severe late side effects. And it would seem like a no-brainer that it would be interesting to look more into this enormous variation in the incidence of complications. I mean, the 0% complication departments obviously must do something right. And you could question whether the 67% complication rate departments did something wrong. And it seems like a study that almost is self-propelling. That paper will almost write itself, won't it? It's all about training and quality assurance and CME and so on and so forth, except that maybe there is another explanation. And I'm showing you the example because I think that's probably what a statistician would do when you go to a statistician to discuss the project. We would be concerned of how much of that variability is likely to be due to differences between the departments in terms of the quality of the treatment they deliver or the case mix that they see. And then on the other hand, how much of it can be explained by chance alone. And this is a plot. Each of the dots now is one of the 55 departments. This is the number of patients they treated in 1993. So that's the number of patients they included in our data set. And you can see that there is a fluctuation in outcome consistent with what was summarized in the abstract of the paper. These are the zero complication departments down here. You will also see this curve. This is the curve of what you would expect if you took a single department's outcome, and you tested that against the national average of 6% severe late complications. This department would have to cross that line for that comparison to be statistically significant. In other words, if we look at the red circles here, the variability we see here is no larger than what we would expect just because of sampling variability. And you can see the trumpet shape. Obviously, the fewer patients you treated, the more likely it is that you would have either a very bad or a very good result compared to the national average, just by chance, without anything being wrong or anything being right, depending on what tail of the distribution you're in. But even that is not the question we should ask, because it looks like 4 of the departments were actually outside that line. So if we tested these against the national average, then we would conclude that they were actually statistically significantly worse than expected. But remember that we do this little exercise for 55 individual departments, and some of them are just bound to be in the good tail or in the bad tail of the distribution. And we can try to adjust for that as well. And that's this stippled curve now. This is simply because if each time you do a statistical test, and if we use the conventional 5% significance level, then of course, if you do that for 55 departments, some of them would probably be statistically significant just by chance. If we redid the whole exercise a year later or 2 years later or 3 years later, we would of course expect, because of what we call regression towards the mean, that these same departments would not come out so bad again. But again, we only had the 1993 data. And the conclusion from this whole exercise was not so much about quality assurance and training, and maybe even volume of patients treated, trying to argue that everybody should be referred to a central center instead of being treated locally. It turned out to be much more a warning about league tables. If you use league tables, and there are very good reasons for wanting league tables of hospital performance, but at the very least, then you need to train the users on how to read the tables and to understand what random variation in outcomes in a given sampling period would do in terms of creating a differential between departments that look fairly devastating, but is mainly explainable just by chance. The only really significant difference outside of the stable line turned out to be Center A. And just anecdotally, Center A was the only center in the UK that actually had a prospective recording of complications in their patients. So probably the reason why they look bad was that they actually did a better job in terms of diagnostic intensity than all the 54 other departments. So a project like this, and it was a lot of fun, of course, being involved as a biostatistician, but a project like this is more about logic. It's not really about computation. These days you can buy a statistical software package, and depending on the test you want to do, you may even find an applet online that will do the job for you. So computation per se is not really a reason for going to talk to a biostatistician. But it's all about logic. It's about using and understanding statistics. It's about challenging your pet hypothesis. And as Richard Feynman said, you should be careful not to fool people, and the easiest person to fool is yourself. We all know what we want to show. Most people don't want to test if A is true. They want to show that A is true. And often it's the role of the biostatistician to say, hey, wait a minute, maybe we shouldn't show that it's true. Maybe we should try to find out whether there are alternative explanations that would be just as likely as the one that we tend to believe up front. Now, this kind of false positive finding from doing multiple tests is an example of what in statistician slang is called the Texas sharpshooter. So the Texas sharpshooter will shoot first and then draw the target afterwards. And then you hit the bullseye each and every time, of course. And it's this idea that the 4 centers in the UK who had a bad outcome, they were not defined before we started. There was nothing simple about them. We simply discovered them, if you like, because they were outliers in the distribution. They looked bad, and that was how they were self-selected. But obviously the outcome was the way we found them. And that is cheating, if you like. I'll show you one more example. I won't go through the details of this in any great detail. If you like to discuss it more, I can definitely help you with that. But this was a project where we used one of the high throughput assays, and I'm including it here just as an example of the issues you face when you have wide data sets with lots and lots of features per case. It's the idea that sequence alterations in the human genome are relatively prevalent. It's around 1 in 300 nucleotides. And with the size of the human genome, that means that we have around 11 million of these sequence alterations or single nucleotide polymorphisms, or SNPs, as they are called. And they are, some of them are fairly frequent in the human population. And they explain a lot of the variability in traits from one individual to another. And of course, it's tempting to hypothesize that they also perhaps explain some of the variability in treatment outcomes that we see after strictly identical treatment for the same medical condition. And these were studies, all of them back in the late zeros. These were studies on radiation side effects. Again, some work that I got involved in. And if you just looked at each of these papers, there were significant p-values everywhere. So people started to study the hypothesis. They now had access to this much more detailed genomic profiling in individual patients. And they simply started to look whether that was associated with side effects after therapy. And yes, it was everywhere. Definitely highly significant. In some cases, there were lots of zeros after the decimal point, and initially everybody were happy. And then we did a study in the UK. We did a validation study. The sample we looked at 92 of these SNPs in 46 different genes. And what was special about the 92 was that they had all been previously reported to be statistically significantly, with a significant p-value associated with radiotherapy toxicity. Back then, this was a fairly large sample, 1,600 patients, 1,000 of them were postoperative radiotherapy for breast cancer, 600 radical prostate radiotherapy. The late toxicity was assessed 2 years after the end of treatment. And this was just trying to argue that we would have fairly high statistical power to actually validate one of these previously identified SNPs if it was not super rare and if the effect size was not very small. An odds ratio of 2.2 would correspond to a patient who, instead of a 10% risk of a late effect, had a 20% risk of a late effect. If it's much smaller effect than that, probably you would never do the test in clinical practice. Assuming an average of, say, 15% would fairly much give a realistic impression of both a 10% risk and a 20% risk case. So that's how we come up with a minimal relevant difference or effect size that we should look for in the study. And now guess what? Not a single one of the 92 SNPs was actually significant. So all of them had been reported as significantly associated with toxicity. And when we did the independent validation cohort, there was not just one, not a single one of them was actually significant. So the literature at that time was all dominated by false positives. The Texas sharpshooter, the fact that if you look at literally hundreds of thousands, or in a genome-wide association study, we would seriously look at in the order of 500,000 SNPs. And then by all means, if you test at a conventional significance level, you would have tens of thousands of these that come out significantly. And obviously you need statistically to address that issue. And there are methods for doing this. This is from a collaboration with Dr. Stas in pathology here at UNB in the biomarker research laboratory, looking at microRNAs in heavy smokers who are being screened with low-dose CT for lesions in the lung. And I don't, again, want to go into the details, but just mention the fact that there are ways, it's called the false discovery rate, where you can try to control the proportion of false positives without being falling off the road to the other side. Some of you have heard about the Bonferroni correction, and if you just do a simple Bonferroni correction, the price you pay for doing that is that you are very conservative. It's unlikely to see even fairly large effect sizes if the number of features per case is large. You simply, you will force yourself to have a much too strict threshold for what you call significance. And the false discovery rate is one of the solutions that addresses this issue. Now, I should perhaps also very briefly mention artificial intelligence. And again, it's something that I didn't mean to discuss in detail. It's a good topic for a separate lecture, I'm sure. But it's also a fact that 90% of the data that is currently stored in digital form is unstructured. And by unstructured, I mean that it is natural language like tweets and Facebook entries or searches on the internet. It's images, photos, biomedical scans, data that is not really annotated. And one of the tools that people are having great hope will allow us to mine some of that data. One of the fields where this is going on currently is in the artificial intelligence field. Natural language processing, for instance, is the idea that you can train a computer to recognize certain natural language constructs, and then you can search through all the data on Google and try to identify incidences of these language constructs, and then try to use them in your research. Robotics is obvious, machine learning is popular, automated learning is popular, reasoning is harder not to crack. Robotics is obvious, and many of you will be familiar with examples of robotics where artificial intelligence will help a robot to do better than if it's traditionally programmed. And you don't need to go far. If you grab your own smartphone, and this is on an iPhone, if you ask for photos of a person, this is an artificial intelligence operation, if you like, and it's fairly successful, no matter the lighting in the room and whether you're wearing headgear or glasses or so on. The likelihood is that it will actually identify photos of yourself with a very high precision. Now, these methods also come with strings attached. And again, like always, it's not about being a believer or a non-believer in the methods per se. It's important to understand that associations are not causal. And again, there are famous examples. This is from a website where you can find some of these nonsensical correlations. This is the correlation between margarine consumption and the divorce rate in the state of Maine. And the correlation coefficient is very, very close to 1. You're probably unlikely to see any correlations as strong as this in your lifetime. And the p-value is highly significant. But of course, it's not super likely that this is a causal relationship. You never know, I suppose. but most likely it's just a coincidence. Now, I wanted to very briefly just highlight another study we did. Again, it's local, it's here. Bill Bain developed a machine learning algorithm for trying to predict 30-day, oh sorry, not 30-day, unplanned readmissions of patients. And again, I just wanted to show you the large sample sizes you end up with, and also maybe highlighting the complexity of such an application, such a research project. This was looking at more than 8,000 features from electronic health record data, and some of these were then engineered, derived from the data, stored, all kind of data, pre-processing work completed, different kinds of models were compared, and so on and so forth. And at the end of the day, you have a model that can predict with some success readmissions across, in this case, 3 University of Maryland medical system hospitals. And where the statistician come in is to say, yes, okay, then we need a validation study, an independent data set. We need to use metrics. We need to have a benchmark. We need to try to decide whether this, although it actually works, whether this actually works better than the more traditional instruments we have already been using for quite some time now for predicting readmissions. And again, that's this kind of thinking. It's not enough to show that something works. You would need to go in and you would show, does it really work better than what we have using traditional non-AI methods, if you like? And also maybe trying to find out what would happen if we implemented this. What would it really buy us? And of course, if you have a program trying to avoid unplanned readmissions, then it's all about how many patients will we need to flag to avoid a given percentage of readmissions. And the efficiency of such a program is not based on the number or the proportion of readmissions you can avoid per se. It's how expensive it is per readmission to avoid it. So say you had an effective intervention, how many patients would you need to take through your flagging and therefore your intervention in order to save a given proportion of the readmissions? Getting all of them would obviously be expensive. Then you would need to target all of the patients, and obviously that wouldn't be cost effective. So again, it's not about the calculation. The calculation itself is fairly trivial. It's all about using and understanding the quantitative methods that will help lifting a project into something that is useful maybe for decision makers or people who were eventually going to implement this or not. Predictors are not causes, and I'm sure you all appreciate this. I think each time we mention this, we need to say it again and again. And because of the complexity of some of these black box algorithms, it becomes a big issue if we can try to open the black box and actually find out what is the likelihood that the classifier that we have been training in our project is actually doing a very bad job. This is a famous example that will be included in a lot of talks like this. This is on the rate of recidivism in defendants in court, comparing black with white defendants. And there's a higher rate of recidivism in black people than in white people. But they don't reoffend because they are black. And again, that should be obvious. It's shown here, if you actually use a risk index, it turns out that the proportion of reoffenders is around the same among white and black people if they have the same risk profile. So there's a lot of focus now in big data inference. In particular, there are other types of AI that are much less controversial. Automation type of tasks are much less controversial. But when you start to train these decision support systems, it becomes a big issue to make sure that it is not unreasonable and unfair. And there are biases. For instance, if you train such a classifier on a patient data set, white patients are more likely to be included, and it has to do with the kind of centers that do this kind of research currently, and they are more likely to have complete follow-up and complete data, and that in itself can create a bias in the trained classifier at the end of the project. So when, that's very quick part of the talk. A lot of the consult starts with the words, "You're a statistician, this will only take you a minute." It all depends, of course, on what you need. Maybe you just need a couple of minutes, somebody who will look at what you've done and say, yeah, that's right, I'll probably do the same. We are a group of 15 currently PhD level statisticians, and we are happy to help you with whatever kind of need, whether you want to do the analysis yourself and just get some advice, or whether you need somebody to actually do the analysis, we will find a way of helping you. So when? We like to be involved from the very beginning. In the problem formulation stage, a lot of projects, if you come too late, a lot of decisions have already been made. Some data will have been recorded in the wrong format. Maybe you didn't put down a date, and maybe we need the date when something happened in order to do the right kind of analysis, adjusting for the time at risk, for instance. And again, you can avoid some of these mistakes, or some of the need to go back and revisit a large number of health records again, if you involve a statistician early on. And we can give advice, obviously, on study designs. We can give advice on sample sizes, data collection tools. We won't collect the data for you, but at least we can give you our 2 cents on what you should collect. And then, of course, we can help you with identifying and actually conducting an analysis. And also, we won't just dump a whole table of regression coefficient in your inbox. We can help you interpreting the model. Maybe we can help you creating some visualization of a model that may be inspiring for people who read your paper, or potentially read your paper, or maybe for future users of your research. So how do you get access to biostatistics support? And again, I'll go through this reasonably fast and leave a few minutes at the end for research. Oh, sorry, for questions and answers. We have an office hour, so once a week you can, without having a prior appointment, you can simply connect and you can get, these days it's online, you can basically walk in virtual space, and you can present whatever kind of question you have. And the trick to get access to this, I'll come back to that a little bit later, is through our admin coordinator, Susan Holt. If you're a cancer center member, then life is beautiful. The Greenbaum Cancer Center supports the biostatistics service by subsidizing cancer-related statistics. Not necessarily 100%. Some people will be asked to pay for at least part of the work, but it's a very good opportunity if you are a cancer center member. The School of Medicine has a system as well. On the website, we have the Center for Innovative Biomedical Resources, and again, by all means, this is a way to get in touch with us. We use the iLab core management software, and you can set up service. Simply, you can set up your project, and that will get you access to a biostatistician who will work with you. And then, of course, we have the Institute for Clinical Translational Research. This is our host today, the UMB ICTR. It's supported by the UMB campus and by the CTSA grant together with Johns Hopkins. The way you apply for biostatistics core support or any of the other core services is through the ICTR website, and the address is down here. And it's being evaluated on a continual basis, so there are no deadlines that you may or may not have passed. You simply submit your application, and generally we are sort of fairly good at getting it initiated and starting to work with you. These are the people involved. A number of us are in the School of Medicine, but we also have a statistician in the School of Nursing and in the School of Pharmacy. And it's really UMB wide. It doesn't mean that if you are in the School of Social Work or if you are in whatever school, you have to find a statistician who already worked with the school. You go through the same kind of portal, and you will meet up. Typically, you will meet up with myself or with Larry Magda, and we will sit down, we will identify the scope of your work and whether it's suited for this kind of support, and then we will assign a statistician who can help you if we decide that it's qualified. Again, Susan Holt is the key person in all this. She will push things forward together with Wanda and the staff in Dr. Davis's office. This is our usage statistics. We have been in existence now for 3 years, from February 18 and until today. This is the number of unique requests. You can see we have an almost linear relationship here, accumulating new projects over time. Currently, the total number of applications when I did this slide was 185. A little less than a third of them are completed. We have 8% that get denied because they fall outside the scope of the program. And then, of course, we have a number that are still in progress. And being in progress means that you had this initial consult. Often people will sit down, we will discuss the project, and they will say, yes, but actually I haven't collected all the data yet. I'll come back to you once I have completed this or that part of the project. And that's absolutely fine. Perhaps I should say that the ball is on your side of the net. You will have to contact us. We will not come back and ask you at intervals whether you are still active or not. That's your responsibility. We will sit and wait for you, doing other stuff, of course, but it's your own kind of initiative to get the service reactivated if it has been put on hold for a while because you were doing other aspects of the study. Predominantly, we have requests from the School of Medicine, close to 9 in 10 of them, but we have from all the schools, basically. And again, this is the graph I just showed you, and we do all of this also in this setting. We can go from just a very short 1 to 2 hour consult and all the way to our standard limit is 25 hours. If it's much more than that, you probably need another mechanism. But occasionally we have projects where we would be on a case-by-case basis willing to go higher than the 25 hours. Types of questions: statistical design, sample size estimation, choice of endpoints, analysis plans, data analysis, preparation of grant applications, consulting in general, and bioinformatics, again on a case-by-case basis. This is what we would call statistical bioinformatics. We don't do the assembly line stuff. You would need to go other places to get help with that, but we would help you linking your high throughput assay data with, say, clinical annotations. So that was the fast overview. Personally, I think this is a great opportunity for people who do need biostatistics support. And at the very least, it's a good way to start getting access to support. Some of these consults will develop into full projects. Maybe the statistician getting support on a future grant application, or maybe finding funds from other sources. But the ICTR is a very efficient way of starting such a collaboration. So with that, I'll thank you for your attention. And as I said, I'm happy to take any questions or comments.
Dr. Bentzen, hi, it's Wanda. Just wanted to let you know that we do have a question in our QA section. Someone is asking, do I need to include bioinformatician in our IRB approval if they are helping us analyze the data from human subjects?
That depends a little bit. It's really a question more for the IRB, I suppose. I think if it's pure assembly line stuff, if it's part of pre-processing the data, and often at that level, it will not be protected individual health information. My guess is that you may not need it, but again, I would strongly suggest that you ask somebody who's closer to the IRB. The IRB we have, we're very lucky, they are very serious. A lot of people have complaints about processing time and so on and so forth. But it's really a great service for all of us. It covers your back, essentially. They try to make sure that all the rules and regulations and human research for very good reasons is obviously highly regulated, and they will help you doing that. A specific question like that should really go to them. And if for some project you think you may not need approval, most likely you do. At least you need a letter from them saying that you get a waiver because the project that you are proposing is so minimal risk that there's no real ethical concern involved in doing it.
Are there any other questions? Oh, one just popped up. I don't think this is probably biostats related, but does ICTR have a service that provides review of surveys?
Gee, that's a question for you, Wanda, isn't it? I think we could help you if you had the data. If you want the instrument pre-reviewed, I think we'd be happy to look at it. I think we'll be happy to give our 2 cents. But you would probably want somebody who's closer to the domain of the research. In a lot of fields, there are established instruments for collecting various survey information from, say, a population of patients. And everything else being equal, we would strongly recommend using an already established scale, maybe even a validated scale, rather than cooking up your own. That kitchen table method often doesn't work so well, simply because there has been a lot of thinking going into creating good surveys already. But there are a number of tricks. If you really seriously want to generate a survey from scratch, I think we can help you to some extent, but you probably need somebody who has domain expertise as well. I don't know if the ICTR per se, I guess there's some expertise in, at least I know some of the involved people who may have relevant expertise, but I don't think there's a core as such.
I'm wondering if perhaps it's a matter of looking to see if the variable names make sense, if there's branching logic needed, if this is going to be a REDCap survey versus a Qualtrics survey. But Dr. Hines, if you would like to send us an email, we can perhaps help you navigate that.
Yeah, definitely.
Now, we have another question here. Do you plan on extending the statistical support services to other UMMS affiliates?
So maybe, Soren, let me...
I'll hand that over to our other panelist. Yes.
Thank you for the question. And the answer is there are discussions ongoing about that very subject. And we will certainly be making announcements and get back to faculty when we have further information. But philosophically, I totally agree with the question. We're very supportive.
So this last question that is on our QA, whoops, nope, not the last one. So nope, especially now, okay. Given the 3 potential ways of accessing support, office hours, cyber, ICTR, how would one prioritize how to access for pre-award support?
I can say a few words about that, and maybe also Dr. Davies wants to chip in here. If it's in the ICTR domain, it's not a bad idea to start through the ICTR, I would say, because you get a fairly effective contact with a statistician who would sit down, and at least you would get going. The Dean's pre-award support is also an attractive opportunity. There are certain strings attached that are not attached to the ICTR support and vice versa. So when you meet with the statistician for an initial talk about the scope of your needs, often it becomes clear if one schema would work better in your case than the other one.
I completely agree, Soren. I mean, the ICTR is disease agnostic, and so across the lifespan. So the advantage there, it isn't, you know, center specific, and so absolutely. And maybe going back to Dr. Heinz's question, I mean, it's twofold. It's, you know, there's skill and expertise need in devising the question, which, you know, content experts, and then there's also sophisticated statistical skill in analyzing the results, and certainly your core would be well served to be able to do that.
Yeah, that's true. And again, if you came even at an early stage, I mean, we would be happy to give you our view on data, say data structure. There are some general rules. In the interest of time, I won't take you through some of them, but there are actually general kind of principles, if you like, that if you incorporate them up front, you do yourself a huge favor later on. There are certain ways of storing data. You may not need to bin them. A lot of, I think, medically trained people like to bin them into intervals and so on. A statistician would say, don't bin them, just put down the actual number. We can do all the binning you want afterwards, but we cannot come from a bin back to the original number. So very simple rules of thumb. Maybe you know them already, but if you don't know them, maybe it could be informative just to sit down with a data person and discuss what kind of advice they would give.
So we have just one more. We already collected data. I guess is never too late to ask a biostatistician for help. Is there a reduced fee for PhD postdocs?
Ooh, now you're getting there. You're putting your finger right into it. Most of what we have discussed today, including the ICTR, requires that you are a faculty member. And if you are a postdoc or a student, you would need your faculty supervisor to be the straw man. And I think we all appreciate that a lot of the work being done in UMB research is done by students and postdocs and fellows. But the person requesting the service must be a faculty person, and that's also true for the dean's support and so on. For the office hours, in theory, you should be on, that's the lowest level of support we can sort of think of. You should, in principle, be faculty also, but we don't turn you away if you are not on the door as a student. We would not leave you outside. We would take you in and at least talk to you. And then we would try to find out how we can use one of the other mechanisms for more comprehensive support. Steve, I don't know, this is probably one of yours as well, but that is the situation, isn't it?
Yes, absolutely. It's exactly what Dr. Bentzen described, and we don't reduce the price because there is no price, and that we give 25 hours, up to 25 hours, and as Dr. Bentzen said, often increased than 25 hours, and that's on the ICTR dime.
It is free, and I think the fact that it is free obviously comes with a kind of obligation. Remember to include it in your acknowledgement in the paper and the grant and so on. Somebody somewhere is paying for it, and we want all the time to be able to document that indeed we are doing some meaningful work and it does add value to research. Again, everybody are welcome. If it really falls outside of this clinical translational heading, we may have to say no for this particular kind of support. We may help you finding another way through the jungle. But if you get support, I think it is really very important. And I'm speaking now not just about the ICTR, but all the core services we have. please recognize, acknowledge. I don't ask you to be grateful, but at the very least, acknowledge that this is something you got and that supported your research.
Okay, I think that's all of the questions we have.
Super, great.
Any
And
closing comments?
Yeah, feel free to contact us and feel free to apply through the ICTR website. We expect to see a blip on that curve of requests over time. We'll see if it actually happens or not. But on a serious note, this is what we do. We like to help you. No problem is too big and no problem is too small. If we can't solve your problem, we will try to give you another problem that we may be able to solve. A lot of what we do is about saying, no, you cannot do that, but you can do something else. Well, why not look at this and that? And again, we like working with you, and it's definitely, it's a lot of fun.
Thank you, Dr. Bentzen. Thank you, Wanda, and thank you, all the attendees.
Yeah, thanks for having me.
Wonderful, thank you. And please remember to complete our satisfaction survey. Again, here's the link, and thank you so much for attending. Have a great day, everyone.
Bye-bye.